Hi to everyone whom are listening to us, to all audiences from worldwide. This is Professor Dr. Kerem Ural from the University of Adnan Menderes, Faculty of Veterinary and Department of Internal Medicine. A mí me gusta hablar español un poco. However, I would like to send my warmest greetings to everyone. En español, me gustaría enviar mis saludos a todos. First of all, for this short, brief explanation of our subsequent study, which is going to be published in Revista MVZ Cordoba, we, along with my teammates, performed a field study for the PhD dissertation thesis of Miss Elif Türk, whom is our undergraduate student, and we performed a field study entitled Assessment of Intestinal Barrier Dysfunction in Neonatal Calves with Diarrhea by using serum, zonulin, and diamine oxidase concentrations. In the study, we thought that one of the major causes of the morbidity and mortality for neonatal calves, and also causing a significant root cause of major economic losses with several different pathogens, diarrhea is our area of interest. Especially disruption of the tight barrier junctions within the especially and consequently disrupting the intestinal barrier functioning and the intestinal permeability. We subjected and tried to make our field study at this era, and we just analyzed two significant biomarkers of both paracellular and also intercellular permeability biomarkers, respectively, such as zonulin and diamine oxidase. Especially by assessing these two different and, to our knowledge, very significant biomarkers, we assessed and have the possibility of interpreting the intestinal permeability. We had performed different groups of calves, both diarrheic and healthy neonatal calves, and we just also made our clinical examination, fecal scoring, and relevant biomarkers investigation. As far as you see from the results, zonulin levels were significantly elevated within the infectious disease of calves in contrast to healthy calves. And the other biomarker, diamine oxidase, it is significantly decreased within the infectious diseases of calves in contrast to healthy ones. And we just analyzed that it was the evidence of proof for intestinal barrier dysfunctioning. So, with the clinical interpretation, we can easily tell that neonatal diarrhea could cause intestinal barrier damage and injury. It also consequently resulted with increased or elevated zonulin and decreased DAO levels, which are in agreement with the increased intestinal permeability. So, with the conclusion, these two biomarkers are quite significant and would hasten also recovery by analyzing its longitudinal analysis. And in the future lab, I think these two parameters should be used for evaluating and interpretation of intestinal permeability. We would like to thank Revista MVZ Cordoba for publication of our study.